very much for coming. It gives me great pleasure to introduce Kay Hodgins from Monash University. Kay completed her PhD at the University of Toronto on the evolution of plant sexual systems, and then she undertook a postdoc at University of British Columbia with Sarah Otto and Lauren Riesberg. And there she used mathematical models to explore the use of selfish genetic elements as a strategy for controlling weeds and invasive plants. She was then employed as a research associate at the same university, UBC, in the Department of Botany, as a bioinformatician for Adaptation, which was a project generating sequence data for thousands of loci across thousands of individuals of lodgepole pine and interior spruce, which are two economically important trees in Canada. She's now a senior lecturer at Monash Uni, where she pursues a diversity of research interests that are generally focused on plant evolution in human altered environments. And she's particularly interested in weedy and invasive plants and uses these species to investigate the genetic basis of rapid evolutionary change. And that's really the topic of today's talk, which is the genetic basis of rapid climate adaptation in an invasive species. So I'd like you to welcome Kay, please.
Hi, everybody. It's quite novel actually getting up in front of people to speak. I haven't done it in a while, so forgive me if I am not as sort of crisp as usual. And I first of all like to thank you, Stephen, for inviting me. It's a great opportunity and to meet people and to share my research and to learn about what's going on here. And I'd also like to begin with an acknowledgement of countries. I'd like to acknowledge the Ngunnawal people, I believe that's correct, as the traditional custodians of the land, and pay my respect to their elders past and present. So I am a senior lecturer in ecological genomics, and I'm really interested in the genetic basis of adaptation and trying to understand that. And over the past several years, I and my lab have become quite interested in invasive species and studying adaptation in these species, both for two sort of central reasons. The first is that we can see and observe evolution happening quite quickly, and this may influence the genetic basis of adaptation. I'm going to talk about that. But also we can have an opportunity to see if evolution in some respects is repeatable and potentially predictable. So are the same genes being used to adapt populations to climate when they're entering these new invasive species are entering these new regions. And so invasive species, they're interesting to study because they impact us, right? They're economic impacts, and they also can have quite substantial economic environmental consequences too. So they're a major driver of biodiversity loss globally. And so there is incentives to understand the mechanisms that contribute to invasion. But from an evolutionary perspective, they're really interesting because they're essentially these natural experiments that allow us to observe evolution happening over short timescales or ecological timescales. So many invasive species, they spread out over really vast geographic regions. In fact, in some people's definition, this contributes or is like a defining feature of invasive species. You see them across really large ranges and encountering quite different climates. So some species have very broad ecological niche. And here's an example of the species we're going to be talking about today. Ragweed, common ragweed, Ambrosia artemisiifolia. It's native to North America, and you can see in its native range, it's found from Florida up into northern Quebec and Ontario. So really vastly different environments. And it's spread all across the globe. It's on every continent except Antarctica. And you can see again in these regions, it's found in really vastly different climates, all the way from the Mediterranean up into Scandinavia and Europe. And it's becoming abundant through Asia, and it's found even here in Australia. Not here, thank goodness, but very allergic to it, but in Queensland and New South Wales. So there's growing evidence, in fact, that actually adaptation to these local climate conditions is an important factor in the spread of some invasive species. So it can be a contributing factor to the success of some invasions. Okay, so one suite of traits, particularly in flowering annual plants, that that are going to be important for local adaptation across climatic gradients are phenological traits. So here, these are transitions between, so they're major sort of life history transitions. So the timing of those, they need to sort of match the local growing season. So plants need to integrate information about their environment in order to germinate at the right time, to flower at the right time, and to set seed at the right time. And if genotypes sort of make errors with respect to their local growing season, that can have very substantial fitness consequences. And across these broad climatic gradients, there's going to be quite substantial differences in the length of the local growing season. So in high latitudes, for example, you tend to find very short growing seasons. So if you think about Scandinavia or Ontario, Quebec, those regions, the growing season is much shorter than areas at lower latitudes, such as in Florida or in the Mediterranean, perhaps. And so we expect latitudinal variation in the length of the growing season, and we would therefore expect particularly these annual species in temperate regions to evolve different strategies with respect to this local growing season. So in areas where there are shorter growing seasons, plants tend to evolve to flower earlier, and this typically comes at the cost to their size. So there's this trade-off, basically, when a plant starts investing in reproduction, it stops growing. And you can imagine that if a plant sort of delays its flowering to grow bigger, and therefore potentially reproduce more offspring, then it's going to get hammered when the fall frosts start, right? So it needs to flower earlier. But in a longer growing season, plants will evolve typically to delay their flowering until they're much larger, and they can invest more resources in producing many more offspring. So you may expect these clines to evolve in flowering time and size along latitudinal gradients, and this is related to the length of the local growing season. So just to give you a bit of background about common ragweed before I move forward, it's wind pollinated, it's self-incompatible. And so these two features basically mean that there's high levels of gene flow among populations. Pollen moves across the landscape. And this is going to be an important point that I'll return to later. So there's very little genetic differentiation among populations in this species. It's annual, so if we're looking to see rapid adaptation, an annual plant is a good candidate. It's native to North America, as I said, and it's been introduced throughout the world, including Europe, Australia, Asia, South America. And it impacts humans. So by every definition, it's a nasty and invasive species. It is an agricultural weed, but its main impact on humans is the fact that it's severely allergenic. So its pollen is the primary cause of hay fever anywhere it's found. In some cases, like say, Eastern North America, Eastern Europe, in the summer, about 50% of the pollen in the air could be from this single species. So it produces billions of pollen grains because it's wind pollinated and disperses its pollen out into the airstream. So there's actually a lot of incentive too to understand the mechanisms that facilitate range expansion from this sort of human health concern. And also the factors that contribute to the length of the pollen season, like flowering time and the size of the plant, which will also influence pollen production. Yeah. So you may think that you're not allergic to it, but if you spend enough time around it, you'll probably become allergic to it, as I found out, unfortunately. So after developing really large common garden experiments, after about a year, I now very, very allergic. I get like a terrible rash on my face whenever I'm around the pollen. So it's a really nasty species, but really interesting, as you'll find out. Okay. So my first PhD student was interested in investigating if these declines in flowering time and size have evolved in this species in the native range, but also in the introduced range. And this could be a signature of rapid adaptation. She looked in North America, the native range, Europe, and in Australia. And so the idea here was to do a common garden experiment where you take accessions from different populations, in this case, all across those three ranges, and you grow them up in a common environment. So you control for differences in the environment. And so if you see phenotypic differences among your populations, then this is an indication that there's genetic differences that are driving those differences in, say, flowering time or size. And if they're related potentially to the climate of origin, this is an indication that potentially climate has been a selective factor shaping that those phenotypes across the landscape. So she did this common garden experiment, taking accessions from across those three ranges. And what did she find? Well, she found exactly what we expected to find, where we see these really strong patterns of trait variation that are related to latitude. Here in North America, you can see that we get a strong decline in flowering time. In Europe, too, a parallel decline, and same in Australia. And this comes at the cost of size here. So plants that flower earlier, they are small, and plants that delay their flowering, they grow much, much larger. And just to give you an idea of the scale of the variation here, here's a couple photos of our early flowering genotypes. These ones are from Germany and Ontario. And these ones, they flowered only after a couple weeks, and they're about this big. So really fast, really rapid flowering genotypes. And then the ones, say, from Queensland and Florida, those ones delayed their flowering by 100 days compared to these little ones. And the plants were bigger than me, right? So the big differences in the common garden. And we can see that even though these populations have only been in Australia for about 100 years, and in Europe for about 150 years, the evolution of these declines and doesn't seem to be associated with population structures, and it's unlikely that neutral processes created these patterns in the traits, especially when we see them evolving again and again upon invasion. And it's not only flowering time and size that we see this in, but there's a whole suite of other traits that appear to be associated with climate, including seed weight, sex allocation, and even secondary compounds. So we see these spatial patterns of variation that are consistent with rapid and repeated local adaptation to climate. And then, the COVID hit, and Melbourne, where I live, we spent a lot of time not in the lab. And so I had to come up with some projects that you could do online, basically at home for undergraduate students. And so I thought, well, there's all these herbarium specimens that are available online. I can get an undergraduate student, and Sam did this, Sam Craig, who's a fantastic student, to just go to these digitized images. And these samples have been collected by botanists over 200 years or more. And just record where the samples are from, when they were collected, the time of year, and the collection year. And look to see if they're flowering or not, or if they set seed or not. So really sort of simple metrics of phenology. And could we see evidence for changes in flowering time and fruiting over time in Europe. And so we found those exactly this. So basically, the probability of observing flowers in these herbarium specimens increased over time, especially at higher latitudes. So more recent collections were more likely to be flowering at higher latitudes. And the same with fruiting. So you can see like later on in the season, we see a higher probability of observing fruits when the accessions are more recent accessions. So this again is consistent with local adaptation of flowering time over time in Europe, particularly at these higher latitudes. So we have some common garden data, so some spatial variation in traits, as well as temporal changes in traits that lend support to this idea of rapid adaptation in ragweed. And these patterns of adaptation in the introduced ranges mirror what is seen in the native range. But I'm interested in sort of the genetic basis of adaptation. So the next question, of course, is, well, can we identify genes that are responsible for this adaptive divergence? And if we see these repeatability at the phenotypic level, are the same or different genes being used to adapt populations of ragweed to climate variation in the introduced ranges as in the native range? And so when you start projects like this, looking at the genetics of adaptation, it's a little bit risky because you could do all of this sequencing and look for associations, but because we're dealing with quantitative traits like height and flowering time, they may have quite a complex genetic basis, right? And so if you think of like classic quantitative genetics, you have the infinitesimal model where you have a large number of loci, each with individually small effects on the phenotype. And if that is the case, you're never going to identify individual loci that are responsible for trait variation, the adaptive trait variation, because their individual effects are so slight. And also, you won't see much repeatability because there's multitude of ways to create the adaptive trait variation across the landscape, right? If the trait's highly polygenic. So you're not going to see, A, you won't be able to identify the loci, and B, you won't see any repeatability. But in ragweed, there are some reasons to expect there to be a significant contribution of large effect loci based on theory. So one is that, as I mentioned before, it's a wind-pollinated outcrossers, and there's very little genetic differentiation among populations. And but we see really strong trait differentiation, so really strong signatures of local adaptation. And so when you have lots of gene flow among populations, if the loci have small effects, then those should be effectively homogenized, right? So selection won't be strong enough to maintain the differentiation among the populations in the face of all that gene flow. But if you have large effect loci contributing to the traits, then those loci will be maintained as different despite the gene flow. And so this may be the case in ragweed. And not only can we think about this in the context of single mutations that might cause large effects, but you can also think about it in terms of clusters of alleles that are found or linked together in the genome through reduced recombination. For example, you can have this with inversions. So when one section is sort of flipped around in some individuals. So, and this actually, inversions typically have reduced recombination within the inversion. And there's lots of theory to show that that can essentially link like effect alleles, and that leads to an advantage in conditions where you have gene flow with heterogeneous selection pressures, because it prevents those beneficial alleles from being broken up with that maladaptive gene flow. Okay, so Another point to make is in the context of invasion, we may expect sort of populations to have been introduced haphazardly into an environment. And the local optima may be quite different from the genotypes that happen to be introduced. So we could think about this as sort of like a sudden change in the environment. And there's a classic paper by Ellen Orr that showed that using Fisher's geometric model, that if you have a sudden change in the environment, large effect loci will tend to fix early on in a bout of adaptation, essentially. And so we may expect in invasive populations, in some cases, to see a substantial contribution of large effect loci that will aid sort of movement of those populations toward that adaptive optimum more quickly. Okay. Another thing that think about repeatability in the context of invasion that would be important is the standing variation that's been introduced to the invasive range. And here's some work done by Lotta, where she used some genetic markers to reconstruct the invasion history using approximate Bayesian computation. And basically, she found two main genetic clusters in North America with some admixture between them, and evidence using these simulations that there were multiple introductions into Europe from these different clusters, and potentially even this admixed cluster as well. So there's no shortage of genetic variation that's been introduced into Europe from North America, potentially including adaptive genetic variation. In Europe, it's a bit, and Australia is a bit of a different story. There's evidence for a single bottleneck introduction about 80 years ago. Okay, so we may expect, given the repeatability in the phenotypes, that we may see repeatability at the genetic level too, because we expect a contribution of large effect standing variants in the invasive range that increase the chances that the same loci, in some cases, will contribute to adaptation in the invasive ranges as in the native range. Okay. So when you do genomics, the first thing you typically want to do is have a good reference genome. So it's very useful when you're trying to do genomics. And so I won't bore you with the details of the reference genome, but basically we've used long-read sequencing, hi-fi long-read sequencing, along with high-C scaffolding to produce a near chromosome-level assembly for ragweed. And here's its, so there's 18 chromosomes, or L90, so 90% of the genome is in 20 scaffolds, essentially. And this is just showing the gene density along the scaffolds and the repeat density here. So in collaboration with Professor Mike Martin in Norway, because we was a student, Vanessa, and pooled our startup money basically, and just sequenced a whole bunch of ragweed genomes from Lotta's experiment and from herbarium sheets. So we have about over 600 genomes, half of them modern samples from Europe and North America, half of them historic samples, most of them are from before 1940 in Europe. Some of them, because in Eastern Europe, those areas were invaded really recently, are from like the 40s to the 70s. And we were interested in seeing if we can use that information in order to reconstruct the invasion history in more detail and to identify signals of adaptation across the genome. So here's sort of a diagram looking at the population structure of the modern samples. Here, just like what Lotta found, we didn't have any Florida samples, but we find two main clusters here, the west and the east, with some admixture between them. And then now this in the Florida, the southern clusters come out. In Europe, we see a mixed bag here, a lot of admixture, some Eastern and Western. But interestingly, we see these single populations showing up as quite divergent from one another. So single clusters here. So just maybe some of you aren't familiar with these plots, but basically each bar here is an individual, and the program assigns genetic clusters to portions of each individual's genome. So that's what we're looking at here. And this is just the genetic clusters that are they're plotted out on a map in terms of the frequency of that cluster in each population. So these are the modern samples. What about the historic samples? Well, we see this remarkable stability in North America and its population structure over time. We get the same clusters coming out in the same general locations. In Europe, a large contribution of this admixed cluster here. And in the historic samples, we don't see those sort of unique genetic clusters coming out like we do in the modern ones. So we see stable population structure in North America. Here's a closer look over time at these bar plots of individuals. You can see the colors are all the same, pretty much over time in North America, but we see this turnover in Europe. And we think this is a function of multiple introductions, a combination of strong selection and drift that may cause this reduction in diversity in some of these populations in Europe. And also we've seen evidence of introgression from other species of ragweed that were introduced at the same time. And this is just to illustrate that we see very little differentiation among populations. So these are FST measures, sort of comparing across the different structure clusters. And their levels of differentiation are very low, except a little bit higher level of FST when you compare the southern cluster to some of the other clusters here. So it's a little bit more differentiated. So this is again consistent with really high levels of gene flow among populations, despite the fact that we see really strong trait divergence over the landscape. So to give you a little bit more insight into the invasion history of the European populations, but now back to the question, what's the genetic basis of adaptation in this species? Well, we've identified phenotypes that we think are important for adaptation across the landscape. Now what we want to do is link that to the genotypes, right? Are there specific regions of the genome that are important for trait variation in traits that are important for adaptation? And so we want to use a top-down approach to examine this question. And the first experiment I'm going to tell you about is a sort of more classic QTL mapping experiment where we took, my collaborator, Romain, took an early flowering German individual and a late flowering Kentucky individual, crossed them, and then produced F1s, and then crossed those to produce F2s. We genotyped them using something called genotype by sequencing. And my fantastic honor student last year did the analysis. And we mapped the reads to our new genome, and we're able to produce this linkage map. So we're looking at markers across our different linkage groups, our 18 different chromosomes, and our recombination rate along the chromosome. So we then take those markers and look to see which ones are associated with our traits of interest in our F2s. In this case, mainly flowering time and height. And so basically what she found was that on linkage group 2, we see a massive QTL. So one flowering time locus explained 23% of the variation in that trait. These are just essentially different measures of flowering time. They all map to this one linkage group 2. We find a height QTL that co-localizes with the large effect flowering time QTL, as well as 2 other minor flowering time QTLs. And we looked at the genes found within these intervals, and we found a large number of different flowering time genes, including flowering time locus T and flowering locus T. FT is very well known flowering time gene. So lots of candidates in these regions. We also then performed a genome-wide association study using the experiment that Lotta did and our million SNPs that we were able to identify through our whole genome sequencing. And this is work done by Paul Batley. It's a fantastic postdoc. It's been driving a lot of the analysis on ragweed. So he used Emacs to control for population structure and then look for associations of our genotypes with the traits all across the genome. And here we're looking, this is a Manhattan plot where we have our SNP positions all across the genome. And now we're plotting the p-value for those association tests for each SNP. And you can see that it was pretty underpowered GWAS. We only had about 120 individuals, but there are some peaks that stand out, in particular this one here. And this peak, these SNPs all land right in a gene that is called early flowering 3 in Arabidopsis. So, and this is a peak is for flowering onset and dichogamy, so temporal separation between male and female function. So a really strong candidate for flowering time variation in this GWAS, and it falls, there's a non-synonymous SNP that falls within this gene. And we plotted that early flowering non-synonymous SNP out on a map, and you can see really striking pattern in both Europe and North America. So the early flowering allele is found in Quebec at high frequencies and in Berlin at high frequencies. So we see this parallel pattern between the ranges with respect to latitude. So this gets to the next approach for identifying genes important for adaptation, climate adaptation, looking for signals of selection using spatial variation. So can we link climate directly to genotype? So there's 2 main approaches to do this. So you can look for extreme differences in allele frequency among populations. So we see alleles that are highly differentiated across our populations. That's an indicator that selection has driven that difference as opposed to drift, especially when we use an approach that can attempts to correct for the effects of population structure. And also we look for correlations between allele frequencies and environmental variables, which can again be an indicator of climate adaptation, especially when they're unusually strong compared to the genome-wide patterns. So we did this using 43 North American populations, 31 European populations, and around 1 million SNPs all across the genome. And we did a windowed approach. So we went along in 10 kb windows and asked the question, do we see elevated signatures of selection in each window? So leveraging information across SNPs, because with divergent selection, we expect it's not just one SNP, even if it's a functional SNP that's going to be diverged, but it's a whole, it's neighbors as well. So we found a large number of candidates for adaptation in each range, and a highly significant overlap between the ranges. So these dark green windows here, those are windows that are outliers in both ranges. So there are climate adaptation candidates in both ranges. So we look for loci that were highly divergent and associated with the same environmental variable in both ranges. So we see repeated patterns of climate adaptation, not only at the phenotypic level, but a significant overlap at the genetic level too. Certainly there's lots of differences, but a highly significant overlap. So it's not a random selection of genes that are involved in climate adaptation across these populations. And there are flowering time genes that are overrepresented in North America, and cytochrome P450 genes were overrepresented as well. These genes are they're diverse functions, including functions in defense, detoxification, and development. So then we wanted to leverage our temporal information, and this is something that is sort of somewhat unique to this data set. A lot of people don't have temporal information, especially over the scale that we have, because few people have access or have sequenced these herbarium specimens or museum specimens at this sort of spatial and temporal scale. So when Vanessa designed her PhD, she tried to identify historic samples that were clustered in time and space, and we picked ones to match modern populations. And so we used these historic modern pairings for each geographic location, and then did scans across the genome, looking for changes in allele frequency over time, as well as reductions in diversity in the modern samples compared to the historic ones. So that would be an indication of the selective sweep when we see those reductions in diversity in windows. So we found many more sweep signals in Europe than in North America, and this is expected because we're looking over 100 to 150 years of evolution, and we don't expect to see that much change in North America, maybe some, but certainly in Europe, we expect to see a lot of change if populations were initially maladapted to their local environment. And that's what we see. So in Berlin, for example, we found sweep signals on the ELF3 gene. So let me just explain this plot a bit. So this is, again, a Manhattan plot, so a genomic location. On the y-axis, we're plotting FST, and this FST comparing the historic sample to the modern samples at that geographic location. And the red dots here, those red dots are outliers in 2 dimensions. The first is this FST, and the second is the diversity ratio between the historic and the modern samples. So ELF3 comes out as a sweep candidate again. And over here, in most of our European populations, we see a giant sweep over here in scaffold 448. 70% of all EU sweep candidates fell in this 14 megabase region in window 44 or scaffold 448. So let's first focus on ELF3 here. So just to remind you, this is our flowering time candidate that we identified in the GWAS. It also showed this latitudinal pattern in Europe and North America. And here we're looking at the change. It's a standardized change in allele frequency from the historic to the modern samples. The distribution is just 10,000 randomly selected SNPs sort of matched for allele frequency to the ELF3 locus. And in Quebec, there's no change over time. Our allele frequency of the early flowering 3 allele, it falls right within the randomly selected SNP distribution. But in Berlin, there were no allele frequency changes from our null distribution that were as great as what we saw in the ELF3 allele for the ELF3 SNP. So this is really strong evidence of selection on this variant in Berlin. So I had a student then who he's become quite interested in looking at structural variation. And so his thesis is called like Beyond SNPs. So he wants to look at copy number variation and inversions. And it can be difficult to look at those types of structural variants if you're just looking at it using short read data, especially in the case as we have here, where it's relatively lower coverage data. But one approach that people have been using to identify structural variants is called a local PCA approach. There's a program called logistically. And basically what it does is it goes along your chromosome and it takes your SNPs, and in each window it runs a PCA. And so it does this window PCA all the way down the chromosome, and then it compares the differences between those windows for the PCA, and then it runs another multi-dimensional scaling analysis on those differences in the PCA structure across the chromosome to identify regions where you have quite sort of divergent patterns of variation. And other people have been using this approach too. It's worked really well in sunflowers, for example, to identify inversions. So if you identify regions that show this sort of divergent signal in their local PCAs, if you then plot out that region on a principal component analysis here, so you just focusing on that one region, if you have an inversion, you expect to see 3 segregating genotypes, essentially, so 3 clusters representing the 2 homozygous genotypes and the heterozygous one in between. So that's what's shown here. And if you take your homozygous genotypes based on the clustering, and we then run FST analysis down the length of the chromosome, you can see that when you hit this region that shows sort of distinct signature in the PCAs, you get really elevated FST. And this is what you would expect if you had sort of 2 segregating haplotypes that were related to an inversion or caused by an inversion. It could be caused by other things that reduce recombination as well, but inversion is a likely cause of this pattern. And we also see elevated heterozygosity in the heterozygous class as well. So Johnny went and he ran these scans across our genome, and lo and behold, he found that there was a haploblock, or at least segregating haplotypes, that align precisely with this region that appears to be under selection in Europe. So we have a likely inversion that is underlying this big change over time in Europe. So then we were able to genotype individuals in the past and the present using this clustering approach. And so we could look at how the haplotypes changed in frequency over time. In North America, there is no change over time. And in Europe, there's been a large shift in the frequency of this haplotypes over time. And again, we looked at the frequency change of the haploblock 448. Here's the red bar. This is how much it's changed standardized over time. And this is our null distribution, where we've taken 10,000 randomly selected SNPs that are matched for the frequency of our haploblock. You can see it's completely outside the distribution, which is, again, is really strong indicator of selection over time. It turns out that about 20% of our parallel adaptation candidates actually fall within haploblocks, or these potential inversions. Yeah, so 27 here, this haploblock actually turns out to be Diana's QTL, the large effect QTL. It completely co-localizes with that QTL. Here we have ELF3, it's found within this haploblock here on chromosome 2. And here's our 448 over here, and there's a few other ones too that overlap with our parallel adaptation candidate windows. These regions are actually enriched for flowering time genes. They're also enriched for pollen recognition genes and immune response genes. So they have genes within them that may think are important for, potentially important for adaptation. This one's interesting. So there's actually one gene that's responsible for 90% of ragweed allergies. So it produces a protein that's found in the pollen. It's found within this haploblock on 27A. It's actually within the height QTL. And this gene has been tandemly duplicated. There's 6 copies of it. Yeah, and it's also shows signatures of selection too. So we looked at the temporal and spatial variation in these haploblocks to get an idea of whether they are changing over space and time in a way that's consistent with adaptation. And here's haploblock 21. We see that in the modern samples, here in green, in both Europe and in North America, there is a association with latitude. In the historic samples, the association isn't as strong, but it's there. And interestingly, we see changes or a strengthening of this association in North America. We might just expect to see it in Europe, but it actually, we see it in both. So we can see the decline quite nicely in the modern sample map here. When we code these into our GWAS, so we look to see if these haploblocks themselves are associated with traits. They are. So haploblock 21, if you are homozygous for the 0 haplotype, those individuals flower later and are taller. And that one's found in the sort of southern environments. This one is Diana's flowering time QTL. You can see this really nice climb here in the modern samples found in Europe and North America. In the historic samples in Europe, we do not see this climb. It actually forms over time. So this is consistent with maladaptation upon arrival, followed by adaptation to the local environment. And just to remind you again that this is the one where in the QTL mapping population explained 23% of the variation in flowering time. And in our GWAS, we see that it is associated with a flowering time trait. In this case, it's the length of the flowering period. So the final analysis we wanted to do here was to try and link the spatial and temporal signatures of selection on the haploblocks. And so we were able to identify the relative strength of selection across the latitudinal climb across the different haploblocks. So if you see a big sort of change in the slope or the frequency across the latitude, that it's consistent with strong selection relative to dispersal. Okay? And basically, the idea here is for haploblocks where we see a really strong spatial signature of selection, those are the haploblocks that change the most over time, which is exactly what you expect with strong selection operating on them. So just to sum up here, so we found evidence at the trait level down to the genetic level of parallel rapid adaptation to climate during invasion. Large number of these parallel adaptation candidates fell within these punitive inversions. We found spatial and temporal changes in the frequency of these haploblocks over time consistent with selection. They're also associated with traits that we think are under selection across the landscape. And they're loaded up with genes that we think could be important for adaptation as well, including flowering time genes and immune response genes. And then we have these interesting pollen genes as well that come up. And I think we've clearly shown that there's a substantial contribution of large effect standing variation that's contributed to rapid parallel adaptation during invasion and ragweed. And so this leads to the final question here is sort of, are there actually aspects of sort of genetic architecture like inversions and large effect loci that actually might facilitate rapid adaptation and range expansion and invasion? So just like to thank a number of people, particularly Paul, Johnny, Chris, Vanessa, and Lada, who did all the work, most of the work. And thank you so much for your attention. And does anybody have any questions?
Thank you very much. That's really interesting. We've got some questions before. Well, Jason and Richard, and there.
Did you say that the ancestral population was Florida in that area? So I was just curious. There was a PCA that you had shown, and the genetic signature from the tropical region was pretty uniform. But at the top, at the north of Florida, there was just a little bit of variation that showed that was correlating with the shorter flowering times.
Oh yeah, so I'm not quite sure where you're saying, but there's no contribution of this southern cluster, this Florida cluster, to the European invasion that we can tell. So
I was curious, where's the ancestral population?
So that's a good question. So I think where you mean in terms of the refugia in North America, because there was glaciation, and then is that what you were referring to, or what ancestral population? Because there's been many, many introductions from North America into Europe. We're talking about the source populations for the invasion.
Right, and the source population is in North America.
Yeah, sorry, yeah. The native range is North America. There's been many introductions into Europe from North America, exclusively from that sort of east, west, and admix zone. Nothing from Florida that we can tell.
Yeah, you mentioned that that was a really good talk, Max.
I think.
And you mentioned that the facilitate invasion and range expansion, but it seems to me like the situation you got here is just things start out maladaptive. So they get introduced from North America, and they're all mixed up. And what's happening is it's essentially reconstituting its North American patterns back in Europe. So I guess I'm curious as to whether you think in this situation here it's just kind of re-liberating itself, or whether you think there's potential for it to actually expand further than what it potentially is in North America.
Well, they actually show, and actually there has been niche expansion in Europe compared to the, I have actually a student that's looking at doing some of this niche modeling too, but in Europe, it was in the Australian context, but there has been niche expansion in Europe. But yeah, I mean, it's a good point. Like it's reconstituting the adaptive patterns that are found in North America in Europe, for sure. But I mean, it's an open question as to, was any of this adaptive variation really necessary for those populations to exist? Probably, in some cases we see like early on that haploblock 21 already showing a climb. So do we see like adaptive colonization happening, at least in some cases where, we probably will, it'd be hard to really address that question. But in other cases, over time, there are changes that we can observe in our herbarium specimens compared to the modern samples. But its effect on population dynamics, that's another question. Yeah.
Question. I had a question. The inversions, are they just happening in the European populations, or are they happening in North America?
Yeah, that's a good question. So they're happening in North America too. And we think they're quite old, and there's some evidence that some of them have actually been introgressed from other species. So we're working on that, and we're trying to date them. Yeah.
So those were the inversions were imported to Europe.
Exactly.
Okay.
Yeah. So here's an example where you see one haplotype common up here, the other haplotypes coming down here, and the same variation's been introduced over here. Yeah.
Yeah, because I wondered if there was any, like the fact that there was so much introduction, or so many introductions into Europe, and mixing of genotypes that wouldn't have occurred previously. And so whether that disruption was causing an increase in the frequency of inversions.
Yeah, maybe. I mean, because we have found evidence of some introgression in Europe that doesn't seem to be present. And so if some of these inversions had their origins in other related species, then, you know, maybe it would be really cool if we found that. I don't think we found evidence for it yet. So, yeah.
Thanks. Yeah, very interesting. I was just wondering whether you've done any look at this along elevation gradients as well, where how that might differ from the latitude gradients where temperature's not confounded with seasonality. And also whether you looked across kind of rainfall gradients, whether there's different things happening with rainfall versus that other access to climate.
Yeah, no, that's a really good question. So aside from maybe in some areas of Europe, there isn't a lot of elevational changes in the, like, yeah, Eastern North America is pretty flat-ish. It's not like, so when I worked on pine and spruce, we could actually take these elevational gradients to our, like, use them to our advantage and try and look for signatures of selection up and down the mountains, essentially. And that sort of minimizes the effects of population structure too, because there's presumably lots of gene flow between those, and you're not going to get compounding effects. But we don't have that sort of same opportunity, I think, in ragweed to tease that apart. But what's kind of interesting, and gets to your question about temperature, I'm collaborating with somebody who has these long, well, evolution experiments in ragweed that they've been running, where they have some warming plots and some control plots, as well as like a biological control too, where they have added the herbivore and they don't have, and they see really rapid adaptation in flowering time and size over only 4 generations. And now I'm really excited because they've sequenced them, and we can see if any of these regions that are evolving with respect to over latitudinal gradients are changing as well. Because they've run a preliminary analysis on a terrible genome that we made a long time ago. And a lot of the candidates that are coming up have like pollen recognition and all these terms that have come up. So we'll be able to pull it all together soon. So yeah, but we want to take a more sophisticated approach instead of just looking at latitude to looking at how these allele frequencies change over time. So we're using some kind of random forest types of approaches to do that. And I should say that when we did those environmental allele associations, we weren't just looking at latitude, we were looking at all the world clim variables. So, yeah.
Alejandro. Okay. I had 2 short questions. One is, do you know of any recognized pathways of transportation between continents? Like, how is this plant being distributed between continents? And the second one is, do you know how resilient is the pollen of the plant to environmental factors? Like, would it be able to survive a trip in a shipping container, for instance?
Yeah, it's not the pollen that you need to be worried about, it's the seeds. And so, yeah, the way that it's gotten across into Europe, and the reason why you have so many introductions into Europe is because of it contaminates like horse fodder. Apparently, like during World War I,
What did you say?
horse fodder, like
Okay.
horse feed. So that's why it really took off after World War I, because of all of these shipments across the Atlantic. Yeah, so that's one mechanism for its movement.
Okay, so just one last question. Sorry.
I had a job.
So my question was about the emergent maxima. Did you look at the expression of this gene between the inversion and non-inversion one, and whether this inversion has affected or silenced or activated something?
We haven't looked at expression in relation to inversion. So the only way we've got looked at the potential function of these inversions has been through relating them to traits, like doing that G was. So we haven't done any sort of expression analysis looking at genes housed within those regions. These are very large regions. They have many genes within them.
Okay, well, I think we've got well, there may